I think we're at an amazing time where we can sample and also detect viruses even before humans or other animals become sick. Virology is a young field as compared to bacteriology because you can grow bacteria on agar plates and they replicate by themselves, and you can open up your refrigerator and you can see bacteria growing. But viruses are very different than bacteria because they can only replicate once they're inside a host cell. And so when researchers started learning about viruses, they had to really think about ways that they could isolate viruses and then detect them. So some of the first viruses that were actually studied and detected were those that could be easily seen by a light microscope. And those include pox viruses, which are some very large viruses. We have had to innovate a lot, and that meant taking primary cells from different animals and learning how to grow them in culture in special incubators, and then being able to inoculate those cells with viruses so that we could start learning about them. So I like to define viruses as very small particles that are consisting of genetic material, so genes, that are protected by a protein-based coat. Kind of protects the RNA and the DNA. They have this nice hard shell and there's something really good inside, and that thing that's really good inside is that nucleic acid. The genetic material that some viruses have is DNA, so that's something that you and I have that makes up our own genomes. Other viruses have RNA, and that's a little bit different. And then there are also viruses that either just have that nice shiny capsid, whereas other viruses will take parts of its host cell membrane and have it wrap around. It ends up having an additional coat. And so what you see is a really amazing variety of viruses when it comes to shapes and sizes, and that is some of what virologists originally used as a way for a basis for naming viruses and for categorizing viruses. We do not know where viruses come from. The field is working on that, and there are a couple of different hypotheses that people like to think about. One is that perhaps the genetic material and proteins kind of got together to form viruses right around the times when the first cells evolved on Earth. Another idea is that viruses evolved from pieces of DNA that are called plasmids. And these are tiny little circular DNAs that sit outside of the host genome. So a disease is usually something that has a symptomology to it or symptoms to it, right? And so there are some diseases that are not caused by viruses or even bacteria. So you can think about things like genetic diseases, for example, that aren't necessarily have anything to do with infectious agents. Viruses, especially the bad viruses, are things that can indeed cause disease. I think what we're starting to appreciate about viruses is that there are some viruses that are probably going to turn out to be beneficial to us. So they're not causing disease, but if we didn't have them, we might not benefit as much or be as robust. So of course there are good viruses, and I'll tell you a couple of examples of how researchers have taken viruses and turned them into something good. So there are a group of viruses called oncolytic viruses. Onco, think about cancer, and lytic meaning to lyse or to kill. And so people have been trying to take normal human viruses and change them just a little bit so that they specifically target and kill cancer cells. There are other people who have used bacteriophage, which are a type of virus that kills bacteria, to lyse bacteria. So we've been hearing a lot about antimicrobial resistance and how there are bacteria that can become resistant to antibiotics. And so phage are one way people are. How viruses can actually be helpful to us as humans. So there's a really neat example of an ancient retrovirus in all of us that produces a syncytial protein, and this protein is important for development of the placenta. And then we're still just now figuring out how viruses of the microbiome can actually be helpful in things like helping us process food and nutrition. That's a special type of virus that needs to use an enzyme called reverse transcriptase, which I know is a mouthful, to make more of its own genome. So a common retrovirus that I think we've all heard about is human immunodeficiency virus, or HIV. There's only a few ways that viruses can spread. With COVID, we've learned about respiratory droplets and how important that is. So if you're thinking about how to protect yourself from respiratory viruses, then you want to think about, covering your cough, or wearing a mask, or sneezing into a tissue and then throwing that tissue away. Viruses can also be spread by the fecal-oral route, viruses that replicate in the intestine and come out in feces, and then people are not very clean with their hands, and they'll then put their hands to their mouths or their eyeballs. So the best way to protect against viruses that spread through that route are by washing your hands or not touching your face. And then, of course, there are viruses that are spread through sexual contact, or sexually transmitted viruses, or sexually transmitted diseases. So we used to think of viruses as very rare things that would happen to us, right? So normally we don't have any viruses except for when we come down with a cold. What we're starting to understand is that actually there are a lot of viruses that are in different parts of our body and at different times. And so there's estimates that there are around 380 trillion viruses either inside or on the surface of our bodies at any time. And so it's really fascinating because we're thinking about trillions of viruses, and when you think about our own microbiomes, you're thinking about billions of bacterial cells. So when you think about yourself as a whole, there's actually probably more viruses and bacteria than there are human cells that make you up. So the way that we've been detecting these viruses is through sequencing of genomes. And the ability to sequence genomes has become much more reasonable, and we've been able to do it from many more locations, and we've also been able to have technology that allows us to detect very few numbers of viruses that are in a sample. And so I think we're at this amazing ability to be able to sample sewage water anywhere in the United States and quickly sequence the material in that sewage and detect if there's viruses or not. People are now working on being able to collect viruses from the air. They talk about the air virome to be able to detect what's in the air. So I think we're at an amazing time where we can sample and also detect viruses at a very early stage, even before humans or other animals become sick. I think the most difficult part of studying viruses is that we only are able to study so few viruses. A lot of viruses that people are finding now, they're finding them through sequencing of different parts of the human body or through the sewage or through the air, but we have no idea what their host cell is or how to propagate them so that we can study them further.